Good morning, everyone, and welcome to today's webinar, hosted by Gator Bio in collaboration with Cactus Bio, entitled Engineering Complex Antigens for the Biopharma Industry, Major Histocompatibility Complexes and Multipass Transmembrane Proteins. My name is Chris Lalu. I am a Marketing Communications Specialist at Gator Bio, and I will be moderating the webinar today. Before we get started, I'd like to go over a few housekeeping items so you know how to participate in today's event. All attendees are listening using your computer's speaker system by default. All the attendees will be on mute by default, and the in-meeting chat option is disabled. However, you can message the moderator at any time in case you're having any technical difficulties. One will have the opportunity to submit text questions to today's presenters by typing your questions into the questions pane of the control panel. You may send your questions at any time during the presentation, and we will collect these and address them during the Q&A session at the end of today's presentation. Getting into the format of this webinar, we'll have both speakers present for about the next 45 to 50 minutes, and once they finish, there will be a quick poll on your screens that we would ask you to participate in, followed up with the Q&A session. So let's begin. I would now like to introduce our speakers for today, Hongshan Li, Strategy Realization Director at Gator Bio, and Manhee Suh, Co-founder and CTO at Cactus Bio. Handing it over to Hongshan, our first speaker of the day.
Thanks for the introduction. Good morning or good afternoon. My name is Hongshan Li, the Strategy Realization Director of Gator Bio. I'm very happy to share Gator Bio next-gen BI technology with you today. First, I would like to introduce the Gator Bio. The headquarters of our company is in Palo Alto, California. Our BI platform technology was invented by Hong and Bob in 2001, and it was called Forte Bio. Unfortunately, there have been not too much innovations on Forte Bio since 2013 due to many reasons. So the first generation of BI basically stopped since then. Fortunately, users found Hong and Bob 5 years ago. They wished they could expand their applications on the BI and hope to get help. Hong and Bob couldn't bear to see the technology they developed by themselves stagnant, so they decided to help. And this is how Gator Bio was founded in 2018. The goal is to maintain all the advantages of the first generation BI, such as simplicity, speed, and ultra-low maintenance, but also has great improvements in all aspects. So the first Gator instrument was launched in 2019. From 2019 to today, there have been 5 new instruments launched within 5 years. So we have more than 50 granted patents. Last year in 2013, 3 instruments were launched within a year, which set up the new record in biotech industry. The speed of innovation of Gator Bio is astonishing. Install base growing rapidly worldwide, and we continue to focus on innovation today. We believe innovation is the future, and Gator Bio is the future of BI technology. Our application area covers biotherapeutics, drug discovery, and gene therapy. Biotherapeutics include quantification, antibody-antigen interaction, affinity screening, and epitope analysis. And the drug discovery and development include small molecule, peptides, and targets binding analysis, enzyme and receptor inhibitor studies, and competitive binding analysis. quantify AAV vectors, detect empty-full ratio, and genome integrity. BLI is the abbreviation of Biolayer Interferometry. The principle is based on the analysis of the interference pattern produced by white light reflected from the surfaces. One surface is the biosensor tip itself, and the other surface is the layer of a biomolecule that deep into the sample forms when the tip interacts with target molecule. So BLI monitors the changes in the interference pattern caused by surface changes. The larger the shift, the more target molecule binding to the capture molecule, providing real-time data on the interaction. I have met hundreds of BLI users and asked them what they like the most for BLI. The answers are rapid and easy, time and cost saving, intuitive software, fluidics-free, and crude samples can be used directly without purification. I believe Gator Bio BLI can provide the best value to our customers. A completely BLI total solution includes instrument, software, and the consumables. If just look at the Gator instrument family, we have 5 instruments today, from 4 channels to 32 channels. The same principle for all the instruments, and the major difference is the throughput. Recently, we have just launched the Gator Pivot, which has the novel evaporation control and the samples cooling. But this feature could be applied to other instruments in the near future. Gator Pro is even more advanced with 32 high frequency channels, which is twice as high as RH96. It's currently the highest throughput kinetics testing instrument in the world. The data quality and the testing time are far ahead of the first generation BLI. Many people have asked me, what's the difference between BLI and SPR? I have put together a table in this slide. We can see clearly the similarity and the difference between these two technologies. BLI is label-free, while SPR requires labels. High throughput assay can be easily done on the BLI at low cost. It uses a smaller samples volume and doesn't require purified samples. SPR is relatively more complicated and expensive. However, SPR software and method may be relatively more mature due to its longer history. Therefore, SPR and BLI are just 2 different methods that can achieve the same result. A manager of the core lab at MIT told me that they have many different technologies in their lab, like SPR, ITC for binding assay. But the most frequently used instrument, guess what, is BLI. Gator Bio's innovation is not just on hardware. Everyone from Gator Bio understands that any successful research invention must solve a real problem. As listed in this slide, in response to users' difficulties and pain points, in addition to many custom-made biosensors, we have successfully developed many fast, simple, and easy-to-use products and testing kits for unlabeled antibody, for labeled antibody and labeled protein, for tagged protein, and for gene therapy. The innovation is never stopped in Gator Bio. Our newly developed Gator 1 analysis software made it even more intuitive to use. We also provide the software for 21 CFR Part 11 compliant. Just to give you a couple of examples for the performance comparison of the first generation and our next generation BI. This is a comparison between the SAXT probe and the first generation BI probe. The data in the upper graph shows that the signal of SAXT is 3 to 5 times higher on average across different assays. The lower graph shows the kinetics experiment of nanoparticle with an antibody. Compared with the first generation BI probe, it overcomes the problem of negative signal. This is another example of the Gator Flex assay kit. The probe can be quickly reactivated and completely remove the strep-evidine biotin molecule complex. A friend asked me what reactivation is and what's the difference between reactivation and regeneration. So the simple metaphor for reactivation is the probe surface is completely wiped out, clear. And then the next run of the new kinetics measurement can be performed. This means the same probe can be used for different proteins and can be used by different users on the different applications. I called it Forever Biosensor, making it very popular in both academia and fully automated screening lab. I also have a great news today for all BI users. Whether you are the first generation BI user or the next generation BI user, both flat and tilt microplate are available for 96 well and 384 well. That means the sample volume can be significantly reduced by the tilted plates, and we make all the plates by ourselves. This means that no longer any plate back order from now on for all BLI users. In summary, Gator Bio's next-gen BLI is one of the best tools on the market for rapid antibody or protein quantification, kinetics, and epitope binding. It's a unique biosensor enabling antibody or protein molecule characterization workflow. Also, it's powerful, easy to use suite for AAV analysis. And we have a wide range of robust BI systems to choose to cover all your needs. The most intuitive software together with fast turnaround for customer biosensor and reliable supply chain made Gator Bio the user's #1 preferred technology platform today. That's my sharing today. Thank you. Now I'll hand over to Manhee for more exciting sharing.
Hello, everyone. Hopefully everyone can see me. Okay, let me adjust the settings. Okay, so thank you for taking the time to listen to this webinar. So we also thank Gator for this opportunity to talk about what we do and about our products. My name is Manhee from Kactus Biosystems. So high quality biological reagents are essential for drug development. This holds true for all sorts of biotherapeutic modalities. So lack of such a reagent, specifically early in the development stage of biotherapeutics, can have a detrimental impact on resources and slow down the drug discovery processes. Today, we'd like to discuss the development of 2 essential recombinant protein reagents that have been previously limiting factor in the research and development, particularly in adaptive T-cell therapy and antibody drug discovery. Those are MHC and multipass transmembrane proteins. So first, our short introduction about Kactus Biosystem. So we are in the recombinant protein business. Our mission is to fulfill unmet market needs for critical recombinant protein for catering extensively to biopharmaceutical industry customers. Our customer ranges from antibody drug discovery to CGT and cell and gene therapy, as well as mRNA therapeutic companies. We are established founded in 2018. Since then, we have served over 1,500 biopharma clinic clients globally. Our team comprises about 200 employees, managing a portfolio of over 3,000 catalog products. Currently, we have 3 different locations. In Shanghai, China, is where we have a R&D and a production and a QC center teams operate. Near 1 hour away from Shanghai, called Lingang, is where our GMP facility is located, hosting up to 1,000 liters of fermenters. And across from the globe and near Boston, Massachusetts, and what is called Waltham, Massachusetts, we have our global operations center overseeing fulfillment and customer services. These are the 4 categories of our products. I'm not going to go details of this. We have notable features like CD3 heterodimers and CD19 and such, but most of our customers actually recognize as a company that tackles a challenging protein, just because we are the first company who actually brought the transmembrane protein called Claudin 18.2 for the industry customers. That protein is a well-known biomarker for gastric cancers. Some of our clients are currently taking part in clinical and preclinical trial using our protein. The second part of the talk will be actually the peptide MHC complexes. Is another topic. We have a growing list of MHC protein, which are essential for TCR-T therapy research. We'll dive into this topic shortly. On the right panel, we have our GMP-grade enzymes for gene editing application. We have a Cas9 max nuclease, as well as a AQ base editor. Recently, we have quite popular demands on the AAV ELISA kits as well. For mRNA therapeutic applications, we also have a GMP-grade restriction enzyme and T7 RNA polymerase that is known to produce far less double-strand RNA that can be problematic for applications. So first topic is a major histocompatibility complex called MHC. This is a very important molecule because adaptive immunity relies on the cell surface presentation of antigenic peptides by the MHC molecules. This molecule displays a set of 8 to 14 residue peptide antigens to T cells. So the interaction between peptide-loaded MHC and TCR provides a means for immune surveillance to detect invading pathogen as well as a developing tumor detection purpose. So because of this development of adaptive T cell therapy, the use of MHC plays a major role. In fact, without MHC molecule, that research cannot be done. But investigating this MHC TCR interaction has been historically quite difficult because the interaction between monomeric MHC and TCR is very weak. So in other words, that interaction is too unstable to be detected. But that all changed. And when 1996, a Journal of Science introduced how to produce tetrameric MHC molecules. So in that study, fluorescently labeled MHC class 1 tetramer complex loaded with the peptide were able to use and to detect CD8 T cells in the flow cytometry. So it was possible not just because of one molecule, 4 molecules MHC molecules basically increase the ability for their TCR counterparts. So this tetrameric molecule enables not only direct visualization and quantification, as well as the phenotype characterization of antigen-specific T cells. So this remarkable this molecule basically transformed our understanding of a cellular immune response since then. However, the process of producing these MHC peptide tetramers is very challenging. To produce MHC molecules, you need to have heavier light chain, and then it need to be refolded from E. coli source from inclusion body, and in excess of a larger amount of peptide. And then separately, you need to produce a tetrameric form that's subtabidine, and then followed by the labeling. So you need to have a chemical labeling process that need to be conjugated together, but MHC monomer first need to be biotinylated, and then finally all molecules come together as a tetramer. So this is quite challenging. So in fact, it's a labor-intensive, lengthy process, taking usually about 2 weeks of processing time. So there is also obvious concern because the whole process involves a complete in vivo processing. So there is a risk of a slight misfolded MHC residual amount of molecule could be presented, obviously. So now, moving past 30 years in our current stage, the process of producing MHC tetramer largely remained the same. Many companies and laboratories stick to this process of producing MHC tetramers. So back in 2018, when we started our company, so we decided MHC should be our long-range project. So that we decided to work on for a long time. And what we want to do is first try to produce dependable and the multifaceted MHC reagents. So what we decided to do is first eliminate in vitro processing involvement. In other words, we would like to produce MHC in completely from in vivo secreted from mammalian cell in a monomeric form. As we are in fact, the first company who are producing MHC in a mammalian system. And then followed by we had a more challenging goal of producing whole MHC tetramer, 4 molecules tetramer, completely labeled with the fluorescently from the cell. So without ever involving any in vitro processing. As a result, we have a list of product here. We'll demonstrate how these molecules operate, performs. The next challenge we worked on was a peptide loading capacity. So in the TCR field, there has been tremendous effort on understanding molecular mechanisms, how peptides are loaded onto MHC group of MHC. Because peptide loading of MHC molecule plays a critical role, as I mentioned earlier, into T-cell response to infectious disease as well as a tumor, but also interaction of maintaining inhibitory receptor on a NK cell, for example. So it is called antigen processing presentation. So it is a mechanism by which MHC are loaded with antigenic peptides. But this is a quite complicated fact that MHC molecules are extremely polymorphic. There are thousands of known human alleles characterized as the HLA A, B, and C classes, and each HLA protein can accommodate over 10,000 peptide antigens. So obviously, a lot of study has been done on this research area. However, not only just to gain the scientific understanding, but also there's a real practical implication of this. It's because there are too many combination of neoantigen can be investigated. That number is way too much to be handled. As I explained to you earlier slides, producing single peptide-loaded MHC takes a lot of time and effort. Suppose 10 neoantigen candidates to be investigated for researchers, which is not a lot of molecules by all means, but all intents and purposes, producing 10 different peptides loaded on MHC is nearly impractical or challenging, or to purchase it itself is very costly. So that is why some have developed tools for different way of doing it. So one way to do it is called peptide exchange. The conditional ligand was loaded and then it has been activated by UV or temperature, or peptide editing method that need to be utilizing molecular helpers, or you can have a mutant, or it's called a B2N orthogonal variance, which is a mutational, such a mutational inducing to induce the polarity, but that method can basically induce incorrect peptide loading. So our approach was completely different. What we decided to do, we say, actually, why can we produce MHC without any peptide at a preloaded stage? But this peptide, free of a peptide MHC, is conformational stable and very receptive to incoming peptide. We decided to call PR-MHC, in short for peptide-ready MHC. So in fact, application is very easy. All you have to do is dissolve peptide and mix with a PR-MHC in excess amount. Within 5 to 10 minutes, you are ready to do the experiment. So there's a huge advantage of doing it. Obviously, save time. It's much easier. Give a researcher for great control over their experiment. The tailored to specific need for their experiment. They can have a different peptide compared at the same time with the same layers of MHC. In other words, also this can be used for high throughput way. Also, it can be applied for many AI computational driven prediction of neoantigen studies. Eventually, those computational studies need to be validated experimentally. So no other way of doing this other than peptide-ready MHC that allows to do high throughput screening method. So as an initial goal, what we decide to do, we believe PR-MHC that is reduced in mammalian cells is invaluable tool for the field of immunology. So how do they perform in here? So showing here is in the middle is called single chain tetramer, SCT monomer. This molecule is basically peptide is physically fused to MHC molecule. Because it's physically tethered, the peptide occupancy is much greater, hence higher sensitivity. We are using this as a gauge for how we perform, how our peptide-ready MHC perform. On the right, we have a peptide-ready monomer without any peptide as a control, obviously no signal. On the left, we have a peptide-ready MHC monomer, simply mix the AFP peptide, and clearly shows that it interacts with the AFC-specific TCR with the equivalent signal to the single chain design tetramer MHC. This is another example showing here in the middle again, single chain monomer with a high sensitivity signal. On the left, on the right, we have a no binding due to no peptide added onto PR-MHC. On the left, we have our peptide-ready monomer. Again, simply mixed with the peptide, and then signal is quite equivalent. These are SPR result using BioRed. On the bottom portion, Gator instrumentation, BLI, basically demonstrating equivalent signal as what we see on the SPIR. And we're grateful Gators actually allow us to test this result different ways. So next slide we are showing is actually peptide-ready tetramer. Earlier two slides we showed how they can be used for monomeric stage. On top row we have a JUCA cell, and the bottom row here is a JUCA cell expressing NY-ESO specific TCR. On the left we have a loaded irrelevant peptide. Obviously there's no signal. In the middle here we have a green fluorescent label. What we call is a FITC equivalent PR-MHC tetramer. Simply loaded with NY-ESO peptide, the light signal lights up. Similarly, we have again APC equivalent, which is a red fluorescent signal. We got a similar result. On the right here is a direct comparison. So one of the vendor who sells a MHC tetramer with a preloaded NY-ESO peptide. Signal reaches about 20% here. On the bottom, we have a APC equivalent. Our peptide-ready tetramer simply mixed with a peptide. Within short minute, we are conducting experiment. We reach basically slightly higher, but they are. So this part of the experiment is called PBMC assay. More specifically, it's called the peptide stimulated PBMC test. So due to PBMC donor variability, we use two donors in this case. Clearly, donor 2 has a much less signal than donor 1. But again, the donor's blood have a different signal, so we had to demonstrate both cases in here. On the right, we are demonstrating peptide exchange method using one vendor B. If you follow the instruction, the whole operation takes about 3 hours. That need to be UV activated, going through the peptide exchange, and their result gets about 46%. And next vendor, if you follow the instruction, it takes about 4 hours operation, and the peptide exchanging reagent had to be utilized, and it shows about 35% signal. On the next, we have our peptide-ready MHC tetramer. Simply mix. Incubation took about 5 minutes. We got the signal of about 45%. Although our signal is basically in the middle, but the whole conclusion here is that we are getting similar result with a minimal efforts. And also, you can actually imagine multiple different peptides can be simultaneously screened by doing this way. On the left here, again, without any peptide, we have no signal. So next slide is a kind of a follow-up few slides, very boring data. This is a stability is a big issue for our customer, biopharma industry. They want to make sure how our products are stable. So showing here is what we decide to do is a HLA MHC, first lyophilized, followed by reconstituted, and add a peptide. And then subject to 3 different conditions. First case is about we keep them 4 degrees for up to 21 days. The next case is 37 degrees for 5 days on the freeze and thaw up to 5 times. Without any peptide, you have basically 0 signal. We are comparing with the single chain peptide, as I mentioned here, is a higher sensitivity marker. Basically, we're comparing to. We have not much differences in activity. It seems to be very stable. This was a monomeric MHC. Next slide is showing you here's a tetramer. Again, in this case, we are taking different approaches. Lyophilize first, reconstitute at the peptide, or we're changing the order, or lyophilize it and then add the peptide, reconstitute, and etc. And then in the outcome result is similar as what we see previous slide. Present thaw up to 5 times, not particularly any differences. Incubate up to 5 days and 4 degrees. So these are the batch-to-batch consistency of products. So on top, we have a MHC monomer, 3 different batch, again, comparing with a single chain MHC. On the bottom, we have a tetramer. On the left, we have HPLC purity. So again, these are the quite, these demonstrate that our peptide-ready MHC are very stable. And then, as I mentioned to you before, the producing different types of allele of MHC in itself take a much long time. Considering requiring many different types of peptide, a traditional way of producing peptide loaded with MHC with a different peptide is not, to our eyes, is not actually a sustainable approach to conduct in the future. So next is a multi-pass transmembrane protein. So these proteins are challenging mainly because of hydrophobic surfaces. These frequently contains multiple hydrophobic helices. They're also very flexible and unstable. So unlike the soluble proteins, these proteins cannot maintain native conformation without the membrane as a part of integral structure. In other words, this shouldn't be taking different approach. So one of the hardest reasons for multi-pass transmembrane is because of low expression, obviously. But there are not only low expression level, there are other consequences of overexpressing multi-pass transmembrane. Of either cell can be toxic, or there can be mechanical disruption of a cell wall. As a result, you will have a much contaminated sample can be produced when you produce a multi-pass membrane protein. So many times, because of these reasons, commercially, many of these products are not available. If there is, very few. It can be very expensive because the cost involved in this method is not trivial. So clearly, there's a huge unmet need, particularly for drug discovery companies targeting multipass transmembrane. So take GPCR, for example. GPCR, most of you are very familiar with GPCR. It's a 7-transmembrane protein. It represents the most important class of drug target. The human genome encodes about 850 GPCR, and half of those are considered drug targets. So given the GPCR's abundance and therapeutic relevance, one would expect to see greater number of drug development efforts, but that's not the case. So number of GPCR drug developed has been low, never been improved over the years. The obvious reason we believe is that the materials, in other words, recombinant transmembrane protein that is active and then structurally sound are not available. That is a primary reason. So to tackle this challenge, we explore 2 different approaches shown in here, either utilizing virus-like particle or nanodisc format. Let me explain you the details of how those involved. So virus-like particle. So over the decade, basically, our company have been actually utilizing this technology and try to improve over the years. So what we are utilizing is a virus nuclei's the ability to hijack host membrane cells, thereby capturing transmembrane protein in their departing particles. So sounds very simple, but it can be very difficult and depending on the types of membrane protein. So advantage of using this method is that multipass transmembranes are in native conformation state within the original host membrane without ever being replaced or extracted by like detergent, for instance. So the clear application, one of the greatest applications for VLP can be used for immunization to get drug antibodies. And we can actually produce many different formats, either fluorescent label or biotin label, which will demonstrate how it can be used. Another means of capturing actual transmembrane protein is called nanodisc. Nanodisc are disc-shaped lipid biolayer, about 14 nanometers in diameter. So this is a synthetic membrane model system that used to be used for studying membrane protein structures. Target transmembranes are basically encased in a lipid biolayer, which is in turn surrounded by amphipathic molecules such as a protein or peptides or synthetic copolymers. So some companies actually extract transmembrane using detergent micelle formats. Obviously, managing this exact amount and types of detergent, as well as a proper condition, can be quite challenging. Not to mention, detergent can actually damage its original structure of a transmembrane protein. On the other hand, conventional nanodiscs is quite complicated, but in fact, the earlier stage of a conventional nanodisc involves in detergent extraction steps. In other words, you have to utilize a detergent to extract the initial membrane protein. Then they need to be replaced with an artificial lipid. And also to remove all this process, you need to use a biobeads. So obviously the list of quite the rare reagent need to be applied. That is the reason why cost of producing conventional nanodisc is quite high. And again, it also involves using detergent. On the other hand, this is a side-by-side comparison. Our nanodisc step, we have developed in a way a method that not utilizing any detergent, any steps. So likely because of this kind of our procedure, that may be why the activity is high and the consistency might be high actually. So another reason why our nanodisc we can produce most cost effectively. So we are actually producing our copolymer, our own copolymer. That is why we can produce a nanodisc much cheaper than a conventional way of producing it. So What you show on here is a basically a basic summary of a VLP as well as nanodisc. In overall, most application overlaps. That can be we can extract using VLP as well as a nanodisc. But major difference here is the VLP we are recommending our customer to use for immunization purposes, and nanodisc can be used for actually screening aspect of this, because nanodisc here, unlike the VLP, does not have any other contaminant protein because it's more purified format. The second application would be depending on the size of the case. So I will talk to you about differences in different types of multipass transmembrane, such as 4 helical bundle or GPCR 7 transmembrane protein, or ion channel and transporter that has about 10 to 14 different transmembrane helices. Depending on the size of a helical format and depending on the cytosol domain, VLP may not be useful. For example, all GPCR and 4 helical bundles can be produced in VLP as well as a nanodisc. But however, ion channels or SLC family protein may not be produced in a VLP if the internal domain is quite large. In those cases, VLP, the nanodisc can be utilized. So first case I would like to share is our Cloudinate 18.2 VLP. So on the left, what you have is a EM image of Cloudinate 18.2 VLP. We have actual physical characterization method. Dynamic light scattering is our common QC procedure as well as HPLC. This is quite more important data. To produce a virus-like particle with biotinylation coated on the surface of membrane, allowing to be captured in a strep-tividin chip on a Bioco SPI instrument in here, and as well as a Gator instrument using strep-tividin probe. So again, both cases, the Gator instrument clearly showed the equivalent result. We thank actually Gator for producing this result. They had, because of the size of a VLP is quite large, Gator people had to use a different software to extracting this data, extrapolating. So that's why we have this result. On the right, we have 3 different batches of Cloud-19 VLP. Here is how this is a method we are detecting and quantifying Cloud-18.1 VLP particle using Cloud-18 CAR expressed in HEC-293. Clearly, you can see no signal here with a CAR. With a Cloud-18 VLP can interact with a CAR molecules on the right here. This slide is quite actually significant. On the left, I'm showing you CD20, which is a 4 helical bundle similar to Cloud-18.2. It's also called tetra-spanning molecule. On the right, we choose to show you one of the GPCR example, GPRC5D, which is important for, I think, multiple myeloma targets. So on the surface, you can see they look all similar in a way that like EM look similar. Obviously, they're all both particle, and you have a ELISA signal seem to be signal, and then you have a good Bioco signal, and FACS can be done both cases. One side you have 4 helical bundle, on the right side 7 transmembrane protein. What we're trying to demonstrate you here is the fact that the virus particle will capture different types of membrane protein. The event or requirement is not the same. For example, to produce or to capture 4 helical bundle, the requirement and the process or the sequence of a timing that had to be occur is different than 7 transmembrane, for example. That is also true for SLC protein. So in other words, I have met some of the customers who have tried VLP in different cases. They have met so many different cases and saying that it does not work. The reason for that is there is a severe consequence depending on the number of helices needed. The outcome of cell morphology or phenotype, for example, can be quite drastic, either cell disruption or immediate cell death or cell basically immediately stop growing or basically suspended movement. We don't know why. We don't have an exact answer for this, but what we are trying to demonstrate here is that each different family of a transmembrane protein requires a different types of a treatment to be able to capture their native structure. So this slide is a immunization application. So on the left, we have a characterization of CXCR for VLP, DLS, again, conventional our QC step. On here, there are customers keep purchasing our VLP, and then we ask them to share their immune response result. We turned out most customers, they never share the results. So what we decide to do, we need to investigate ourselves. What are the effective dosage range that need to be used to get good antibodies? These are our in-house results. So as expected, as we know, VLP is a very good have immunogenicity. So that's because VLP has the ability to deliver antigen in a dense and repetitive manner, allowing B-cell receptor to cross-link effectively. That's why you can get a very good antibody out of it. So in here, what we're showing here is that if you had any amount of VLP, including 0.5 microgram of VLP, you will have a strong immune response. On the bottom part of the figure, what we're trying to analyze is, okay, what is the effective dosage range that was enough that we got a good antibody? The answer is also great. It's very broad. In other words, if you have no VLP, no signal, even 0.5 microgram VLP, you have some kind of immediate signal. 5 microgram VLP, you have a very strong signal, and again, basically reaching to the same plateau as the rest of the VLP, including up to 100 microgram. This was analyzed using CXCR for a nanodisc, particularly purified for this analysis. So the results is very clear. We are comparing here well-known drug antibody from a patent, CXCR for antibody, comparing with our anti-serum out of this result. You can see almost all antibody what we have in the serum is a very strong, very good antibody. As a result, actually, we were able to analyze and identify various good CXCR for antibodies, including the well-known ligand binding capability here. So this is a CCR8. This is actually controls the immunosuppressive function of a tumor infiltrating Treg cells. This is very difficult to produce. Main reason is the moment you express CCR8, cell dies immediately. So we had to go through quite a trouble how to capture the right moment and then how to suspend that in the outcomes to be able to capture. On the bottom, we have finally successfully produced a CCR8 VLP, and we have an analysis result. And showing you here is a nanodisc analysis comparing with a vehicle. And after we do the immunization, we got our own antibody. What we realized is that our antibody against a CCR8, human CCR8 antibody, VLP cannot bind well the signal. The reason for that is the N-terminus domain, somehow it does not bind the signal. So as most of you know, cross-reactivity of antibodies is quite important for preclinical application. For that, what we decided to produce dual CXCR4 VLP, meaning it has a human CCR8 and cyno CCR8 on the same VLP surface, and demonstrate here amount of population is very similar. Currently, we are undergoing this processing. We have a strong indication that likely this approach will result in the antibody with a strong cross-reactivity of the different animal species. Obviously, we can do that with our mouse or cyno or any other animals sent together. So we are even to be able to produce triplet VLP as well. So this is the VLP sorting. We all know cell can be sorted out for fax machine. So what we are trying to envision in this case is we are trying to see VLP as a small form of cells with the enriched target multipass transmembrane. So what we have here is a overly expressed CXCR for VLP, which is a green fluorescent label. When you have a antibody, the APC label antibody, you can see quite a shift. Similarly, you have another case of VLP, a CXCR for VLP. When we have a CCR antibody fluorescent, you can see quite a binding, the same for CCR7. There's another example of another GPCR, MRG PRCX VLP. When you mix with antibody, you have a clear shifting. So this allows some of the cases where study cannot be done. For example, one is to investigate a toxic GPCR, such as, let's say, CCR8. We cannot possibly produce on a cell membrane and try to do this kind of assay. If we were able to enrich CCR8 on a VLP surfaces, and that can be sorted out, and that can be used for characterizing your antibodies. So following few slide is our customer allowed us to share. They are actually sharing their result. This is a Galangen, and it's a San Diego-based biotech. They actually use our both nanodisc and VLP GPCR, and they were very graciously allowed their result. And according to their result, our both nanodisc and VLP can be used for yeast display applications. Next slide. This is Adapt Therapeutic in Boston-based biotech company. They have been working on A2AR in the first in-class antibody drug they are working on. We have a VLP, and we also produce nanodisc. In their application, we were trying to produce a nanodisc that can be used for FACS sorting. Unfortunately, what we learned is that our copolymer seem to be incorporated into the cells. So therefore, we can see control CCR7 and A2AR nanodisc. They seem to be not actually applicable for FACS usage. So that's why we engineered biocompatible nanodiscs that can be used for FACS sorting. So here you can clearly see control have a no signal. Biocompatible nanodisc A2AR have a strong signal recognizing A2AR displaying on the HEC293 cell. So this is another family protein, SLC transporter. As name implies, this transports mostly very small molecule and nutrients and neurotransmitters or drugs. This has been recently recognized as a key biopharmaceutical targets. Unfortunately, this class of protein has been understudied. But we also realize that this kind of a SLC transporter cannot be well sustained in the VLP format. That's largely due to cytosol domain is way too large. In other words, encapsulation, it will be conflicting with the encapsulation part of capsid part of a VLP. So however, we are able to produce all the following lists shown in here in a nanodisc format using Gator instrument. One of the SLC 13A5 nanodisc seem to demonstrate a very good activity. So in summary, we are offering many different options to our customer with a very affordable rate in the many choices of our multipass transmembrane. For one, we can produce a nanodisc format with a polymer biocompatible cases. We can biotinylate, or all this can be used as ELISA, SPIR, BLI, except for polymer, it should not be used for immunization or FACS in our opinion, but biocompatible can be used for all. The VLP, likely we strongly recommend that can be used for immunization. As I mentioned earlier, none of this our multipass transmembrane ever used or involved in any detergent usage, unlike a conventional nanodisc format. In terms of production-wise, what we are trying to make a statement here is that we can produce all different types of transmembrane protein. We strongly believe that multipass transmembrane is no longer a limitation for drug discovery, because we can produce foilic bundle, also known as a tetraspanin, or GPCR or SLC protein that containing up to 10 to 14 transmembrane. Either we can produce a nanodisc format or VLP. These are the list of the GPCR we're currently working on. If you don't have a list in our website, you can personally, or you can contact us ourselves, and we can produce within the 4 to 6 weeks period of time. Turnover is pretty quick. For that, I thank you for listening to our talk. We'll take more questions.
And now we're going to begin the Q&A. So thank you, Hongshan and Manhee. We're going to begin answering the questions submitted during the presentation. As a reminder, you can still submit questions through the questions pane in your attendee control panel. Okay, so it looks like we have a question here. I think this is for Manhee regarding CCR-8 nanodisc. Do you always see KD values from SPR and BLI this close?
Usually, yes. I think don't get me wrong, I'm not an expert on neither of those instrumentation, but as I understand and our observation, when we get the both result, and then BLI signal seems to be a little bit lower than SPIR. I'm not saying it's good thing, bad thing, but overall result is very comparable. That's what we see. The numbers are quite close.
Okay, thank you.
I mean, what I mean is very small. It's not a huge, like a multitude of differences. Slightly lower. Yeah.
Okay, great answer. I see two more questions here popping up right now. Has BLI been used to study GPI anchoring? Could nanodiscs be used to functionalize the biosensor, et cetera, to study GPI painting?
My first question is, what is GPI? I'm not that familiar with. It's a type of protein.
Let me read it one more time here. Has BLI been used to study GPI anchoring?
Oh, I see. Okay. That I think Gator people should answer.
Glycosylphosphatidylinositol. I can't
Yeah.
quite pronounce it. I
Yeah,
apologize.
actually, I happen to know that portion. So actually, we have one of our R&D's exploratory research we've been doing is to try to capture those molecules on a VLP. Actually, we can capture the membrane, in fact, but I don't know if that can be utilized on the BLI is another story. But in terms of how we can put the modification we're talking about, it is a very basically single residues. I believe it's a serine residue or glycine, something like that, that is anchored to the membrane, the lipid, basically. That can be done in VLP.
Hongshan, could you also answer that first part? Has BLI been used to study GPI anchoring?
So it's a case-to-case different. So definitely worth to try. So
Okay.
I would love to try. So if it works, great. So in theory, it should work.
Okay, I'm gonna move on to the next question here. This is from Steve Molinarola. Hey Manhee, you mentioned the copolymer nanodiscs had background interaction FACS and specifically mentioned this as integration into the cells. Do you have data to support the integration or just interaction to the cells?
Oh, actually, we do have the proof of doing that. Actually, I happen to be, I mean, we knew that actually occurs, but I happened to be sitting in a PAX meeting last year, and then I happened to, and I think at a FABS meeting, I'm sorry. So there is what they called an nanodisc fusion or something like that. You can actually, having nanodisc with the copolymer can be actually fused onto the cell surface, actually reintroducing. So that event is not just what we just observed. Actually, that can occur and you can use to actually label the cell surface, actually. That event is well studied, currently well investigated. In fact, our company have produced nanodiscs for that purpose so that we can actually reintroduce the fluorescence signal, for example. So copolymers, the main job is basically it actually maintain the phospholipids surrounding it instead of using membrane scaffold protein, for example. So it is well understood that it can actually penetrate into the cell membrane.
Thank you, Hongshan. And I think this one may also be for you, Hongshan. It says, any lipid or hydrophobic specific biosensors?
Oh, yeah, we have some custom biosensors you can try. And so if you have any like specific lipids or nanoparticles you like to run on Gator Bio's BI system, just let us know, and we can work together. And we have developed the software kind of matching that. We would love to work with you for that kind of application.
So as just a kind of a comment for this, actually the Gator has been helpful for some of our data where we have a trouble to get, particularly VLP, for example. And then signal is high, and then technical support was great, and they actually troubleshoot to be able to extrapolate the data we had that could not solve anyway. But they can be very good help. Or that I can just recommend them, other people, yes.
Love to work together for any new applications.
All right. Thank you, everyone. And Manhee, it looks like we have a few people requesting to connect with you. Just so you all know, this webinar is being recorded. So I'll be sure to share this with you later, and I'll pass any connections forward. So at this point, I want to go ahead and end the Q&A, but if you have further questions, be sure to email us. So thank you to our speakers, Hongshan Li and Manhee Suh. Thank you everyone for attending today's webinar. And if you have any other questions, please contact us at info@gatorbio.com. Once you leave today's webinar, you will receive a follow-up email within roughly 24 to 48 hours with a link to view the recording. It'll also be on social media, of course. So on behalf of Gator Bio, Kactus Bio, and our presenters, thank you for joining us today and have a great rest of your day.
All right. Thank you, everyone.
Thank you.